go to the original mother strand. So this is the summary of the DNA replication, the entire process. So as I mentioned, okay, the two strands, they are connected by hydrogen bond, okay. And then this is the second enzyme I want you to memorize, DNA helicase, okay. We know that DNA is in a double helix normally. In order to replicate the DNA, we need to unwind the double helix. So this enzyme, DNA helicase, is the one that unwinds the double-strand DNA. So the function is also written on the name. ASE is the enzyme. What does this enzyme do? To unwind the DNA double helix. The helix is the DNA double helix. Is to unwind DNA double helix. So and then after DNA helicase unwinds the DNA, then the two strands, they are separated, so that nucleotide, new DNA nucleotide, I should say new DNA nucleotide, can fit into the separate strands, forming the complementary base pairing. So what it means here is that after we unwind the DNA and split the two strands, then you can form the new complementary strand. That is the green one that I drew in the previous slides. So when nucleotide fit into the mother template strand, then the new green strand will be formed. Which enzyme do that? Which enzyme add the new DNA nucleotide to form the green complementary strand? It is done by the DNA polymerase that we just talked about in the previous slide. That's why I asked you to memorize the first enzyme, DNA polymerase. It makes DNA polymer. How does it do it? By fitting the new DNA nucleotide to the mother strand or the template strand, so that this new DNA nucleotide, they will form the complementary strand, which is the green one that I drew in the previous slide. Let's continue the process. This is the last enzyme that I want you to memorize, DNA ligase. So the function of DNA ligase is to seal. You know, when we put the DNA's nucleotide to form the complementary strand, sometimes the complementary strand, okay, how should I put it? Okay, let's show it here. So you have the DNA here. The mother DNA is now split into two. So one strand is 5' to 3', remember? One strand is 3' to 5', remember? Now, so first of all, you have the DNA helicase sitting here. DNA helicase sitting here, because DNA helicase move up as it moves up, it will unwind the DNA. Okay. And then DNA polymerase. You will have two DNA polymerase. Let me see. Okay, I'm lazy. I'm just put P. You know that this is polymerase. DNA polymerase will move from 3' to 5', this direction. And then another DNA polymerase will also move from 3' to 5' direction. It is also a DNA P. Okay, let's add more. As the DNA polymerase move from 3' end to 5' end, what will happen? Oh well, I shouldn't use red color. I shouldn't use red. I shouldn't use green. Okay. Remember, you make the complementary strand. Okay, but the complementary strand, the new DNA strand, it is always made from 5' to 3'. So this is 5' here. This is 3' here. So this is how what during the DNA replication what happened. So the two double helix molecules are identical to each other and to the original DNA molecule. Well, we already keep repeating this. Point number 5. Okay, this is the overview. What they show here is basically what I drew previously. Let's take a look at the details. Well, you have the Helicase here. I'll just put H. Okay, Helicase is here. It is moving up. As it moves upward, then you keep on unwinding the DNA, okay? And then you will have the DNA polymerase. Okay, let's pay attention. The mother strand, it is 3' here. So in other words, this should be the daughter strand. Here will be the 5' end. If the daughter strand is 5' end, then the DNA polymerase here. Okay, this is polymerase. Will be moving to this direction. In other words, as the helicase keep moving up to unwind the DNA, polymerase would be following the helicase. to elongate this complementary strand. And this is the 3' end here. For this, we call it original template strand. We call this the leading strand. The leading strand because helicase is leading the polymerase. Helicase is leading the polymerase. So that you can see that as the polymerase make or elongate the green template strand, the green template strand will be continuous. It's like one long straight chain, okay? The issue is on the other chain, on the other strand. As you can see that the other mother strand, this is the 5' end here. Okay, this 5' end, what it means is that the complementary strand here, this is the 3' end here. Okay? And then this is the 5' end for the complementary strand. So where will be the DNA polymerase? DNA polymerase will be around here. This is the DNA polymerase. What is the direction of movement of DNA polymerase? It moves downward. Because as I mentioned that the complementary strand is always made from 5' end to 3' end. The green complementary strand is always moved from 5' end to 3' end. So what happened? What would be the issue? The issue is that as the DNA polymerase moved from the 5' end to the 3' end like this, as it reached the end of the original template strand, then what does it need to do? Then it will go back to the original fork of replication. This is called origin of fork of replication. Then you'll go back here, and then this is the P. Okay, this is the P, the polymerase, and then you will continue to replicate along this direction. Oops, now I should put it here. So it will replicate. So the replication, the DNA polymerase, the new complementary strand will be like this. You will form here, 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 and here. Okay. In other words, it will make a 5' end here. Okay, I draw it too big. Okay, let me erase this. It's too big. So this is the 5' end here. The second then the 3' will be here. In other words, you will see two green complementary strands, two separate complementary strands. Two separate complementary strands. Okay, this one is the first one. This one will be the second one. So you have two separate complementary strands, and there's a gap in here, right? The gap in here will be sealed by, the gap here will be connected by the third enzyme that I want you to memorize, DNA ligase. This is what DNA ligase do. It connects the two separate complementary DNA strands. So DNA ligase will seal these two, the strand 1 and strand 2, the purple, the one that I indicate in purple, strand 1 and strand 2 together. So this is what DNA ligase will do. And as you can see that when the DNA polymerase goes down, move down to make the number 2 green complementary strand, when it reached the beginning of the 5' end of the number 1 of the complementary strand, what will happen? Then it will do the same thing. It will go back up until because the helicase keep moving upward, right? The helicase may now go to here. Now the helicase may move up to here because it keeps moving upward. Then when the DNA polymerase bind to here, then you will just move down to continue for the replication. So basically it just keep on jumping. It goes down and then jump back up and then move down and then jump back up and then move down and jump back up. So it keeps doing this, so that you will have a fragment of the green complementary strand. You have different fragments. And then these fragments, they are sealed or connected by DNA ligase. So that's the whole purpose of DNA ligase. So the mother strand that have this kind of a fragment of the green complementary strand, we call it lacking strand. As you can tell why we call it lacking strand, because the DNA replication is lagging behind. So for this original template strand from the mother DNA, we call this is the lagging strand. So this refers to this one here. This refers to this one here. Okay, so the mother strand, one strand will become leading strand, the other one become the lagging strand.